This is week 12, Molecular Genetics and Human Disease, Part 2. Duchenne's muscular dystrophy is one of the most common neuromuscular disorders in children. It's an X-linked recessive disease. The absence of the protein dystrophin, which is critical for the maintenance of muscle, results in generalized muscle weakness that progresses over time. The clinical manifestations usually occur before the age of 6 and can emerge in infancy and include fatigue, delayed walking, difficulty climbing stairs and running, an enlarged appearance of the calves, which is referred to as pseudohypertrophy, and a Gowers sign in toddler years, which is the inability to rise from the floor in a normal fashion. This is a red flag for the disease. Please look at these two videos on the Gowers sign. So why pseudohypertrophy? Well, if you take the name apart, pseudo means false and hypertrophy means for the muscle to grow and to be larger. And in this case here with the Duchenne's muscular dystrophy, what is taking place is that the calves are appearing larger due to the replacement of normal muscle tissue with connective tissue and fat. As a child is growing, the disease is progressing, and in the teenage years, the child loses the ability to ambulate, and this has an impact on the cardiac respiratory muscles. The DMD diagnosis is made through clinical symptoms, through a muscle biopsy, where you would find the absence of dystrophin, elevated creatine levels. This is attributed to muscle wasting. An electromyography in EMG can be done for diagnosis, as well as DNA testing for the gene. Death usually occurs from pulmonary complications and/or congestive heart failure in the late teenage years or early 20s. Treatment involves maximizing the quality of life with corticosteroids promoting mobility and emotional support for the child as well as the family. Becker muscular dystrophy, BMD, is due to mutations in the dystrophin gene, and you can generally find dystrophin demonstrable in the muscles of patients with this disorder. It is X-linked recessive, and the BMD alleles cause a much milder disease. In fact, patients are said to have BMD if they're still walking at the age of 16. There's significant variability in the disease, and many of the patients remain ambulatory for many years. The mutated allele that causes this disorder can maintain the reading frame of the protein and thus express some dystrophin and an altered product at reduced levels. If we look at reproductive fitness and we compare DMD and along with the BMD, we find that the DMD is a genetic lethal. 1/3 of the cases of the DMD are predicted to be new mutations, and 2/3 have carrier mothers. So the reproductive fitness of males with BMD is high. It's about up to 70% of normal. And there's a high proportion of BMD cases that are inherited and only about 10% that represent new mutations. Let's look at the carrier females of DMD for a moment. The great majority of the female carriers have no clinical manifestations. Approximately 70%, though, have elevated levels of creatine kinase. 20% of adult female carriers have muscle weakness, and 8% have life-threatening cardiomyopathy and serious proximal muscle disability. Only rarely do women have DMD. Please go to these websites to learn more about muscular dystrophy. I'd like to go back and to remind everyone about penetrance and expressivity and the fact that the same genotype can produce different degrees of a phenotype in different individuals because of influences of other genes, as well as environmental influences such as nutrition, exposure to toxins, and to stress. Huntington's disease. Huntington's disease is a progressive brain disorder caused by a single defective gene on chromosome 4. It is an autosomal dominant disorder. It is also known as Huntington's chorea, and it is named for George Huntington, the physician who first described it in the late 1800s. If you'd like to look at some interesting stories about the pilgrims who came over on the Mayflower and their descendants, and the stories behind the fact that some of those descendants may have been executed at the Salem witch trials due to the way that they were acting, being attributed to Huntington's disease. Huntington's disease is one of several trinucleotide repeat disorders, which are caused by the length of a repeated section of a gene exceeding a normal range. The gene is located at 4p16.3. It contains a sequence of 3 DNA bases, cytosine, adenine, guanine, and CAG, which is repeated multiple times. CAG is the 3-letter codon for the amino acid glutamine. The production of a chain of glutamines is known as a polyglutamine tract or polyQ tract, and the repeated part of the gene is the polyQ region. Trinucleotide CAG repeats over 28 are unstable during replication, and this instability increases with the number of repeats present. Usually, this leads to new expansions as generations pass, instead of reproducing an exact copy of the trinucleotide repeat. What happens then is this results in the number of repeats changing in successive generations, such that an unaffected parent with an intermediate number of repeats, 28 to 35, or reduced penetrance, 36 to 40, may pass on a copy of the gene with an increase in the number of repeats that produces fully penetrant Huntington's disease. Increases in the number of repeats and hence earlier age of onset and severity of the disease in successive generations is known as genetic anticipation. Instability is greater in spermatogenesis than in oogenesis. Maternally inherited alleles are usually of a similar repeat length, whereas paternally inherited ones have a higher chance of increasing in length. It is rare for Huntington's disease to be caused by a new mutation, where neither parent has over 36 CAG repeats. In individuals who have Huntington's disease, they demonstrate uncontrolled movements, they have personality changes, they have brain damage, and the total mechanism is not understood at this time. The symptoms usually begin between 30 and 50 years of age, but can start at any age. The disease may develop earlier in life in each successive generation, as I previously mentioned. If inherited from the father, the disease manifests earlier, and 10% of individuals who have Huntington's disease are under the age of 20 and have juvenile Huntington's disease. I have included a link to a video on Huntington's disease of the juvenile form. Some of the complications in Huntington's disease are pneumonia, heart disease, and physical injury from falls, which all work to reduce the life expectancy. Suicide is the cause of death in about 9% of cases, and death typically occurs 15 to 20 years from when the disease was first detected. Woody Guthrie was a folk singer. His full name was Woodrow Wilson Guthrie. He was a singer-songwriter and one of the most significant figures in American folk music. He lived from 1912 to 1967. He was diagnosed in 1952 with Huntington's disease. He had a total of 8 children, and 2 of them had Huntington's disease. A few of his children died young, so there might have been others who might have developed it at a later time. If you'd like to follow this link, you can go to Ken Burns' story about the Dust Bowl, and you can hear Woody Guthrie sing, and you can hear a little bit about Woody Guthrie in the narration. Please look at these videos on Huntington's disease and Woody Guthrie and juvenile Huntington's disease, and then also read the Thoughts for Genetic Counselors stories and snippets shared from members of the NYA. Marjorie Guthrie married Woody Guthrie in 1954, and they had 4 children together. And after his death, she founded the Committee to Combat Huntington's Disease, which eventually became the Huntington's Disease Society of America. This is the poster to commemorate 50 years of service, 1967 to 2017, for the Huntington's Disease Society of America. You can see in this picture Woody Guthrie and his wife, Marjorie. Please go on now to the case study.